Hi, my name is Matthias Fahrner, and I'm from the Institute for Surgical Pathology in Freiburg, Germany. Today, I would like to talk about democratizing DIA analysis on public cloud infrastructures via Galaxy. First, I would like to introduce some of the principles and characteristics of data-independent acquisition mass spectrometry. Generally, this includes normal label-free sample preparation, during which the proteins are solubilized, digested, and most often cysteine residues are alkylated. Cleaned up samples are measured in the mass spectrometer, leading to mass spectra. One of the most commonly used methods in mass spectrometry is to select precursor from the most abundant signals in the MS1 and target those specific mass over charge values for a second scan. Prior to this second scan, the selected precursor undergo fragmentation, leading to precursor-specific MS2 spectra. Due to the intrinsic dependency on the most abundant signals in the MS1 scan, this is also referred as data-dependent acquisition. DDA data can be analyzed using a variety of software, for example MaxQuant. During the last decade, another method emerged called data-independent acquisition. Here, the MS2 scans are predefined and fragment spectra containing all precursor within the respective mass over charge window are collected and fragmented. Just as for the DDA approach, there is a broad variety of software which enables the analysis of DIA data, for example Spectronaut, Skyline, and OpenSpot. Let's look at the DIA method in more detail. First, a wide range MS1 scan is performed, followed by a large number of MS2 scans covering the same mass range. In this example, the smaller windows are all 25 Thomson, ranging from 400 to up to 1200 Thomson. After another MS1, the sequence of MS2 scan is repeated, leading to a series of MS2 scans of the same mass over charge range. For quantification, the intensity values of the consecutive MS2 spectra are integrated over the measurement time for each M over Z value. To investigate the two different methods, we measured 12 replicates of T lymphocyte samples. For both methods, the number of cumulative protein groups, meaning proteins which were identified in all runs, are decreasing with additional runs. This effect is stronger visible when looking at the identification of peptides. In general, DIA shows higher number of cumulative peptides and proteins across multiple runs. This highlights the reproducibility of the measurements and enables more comprehensive comparison between the runs. However, using DIA, one also needs to overcome certain challenges during the data analysis to account for the intrinsic characteristics of this method. Due to the broad isolation window on the mass over charge axis, the MS2 spectra contain fragment ions coming from multiple precursors. This complicates the correct identification of all present peptides. Also, there is no direct link between the single selected precursor of the MS1 and the corresponding MS2 spectra. Originally, the data analysis of DIA data requires a priori information, for example spectral libraries containing the possible peptides and the corresponding fragmentation pattern. More recent efforts allow for using predicted spectral libraries, which have been shown to allow for decent identification results. Due to the complex data and multi-layer analysis, often multiple software is needed for a complete DIA analysis, thus complicating streamlined and straightforward data analysis. Here we can see an example of a classical DIA analysis workflow using open source software. The different color indicates if the analysis steps are performed using a graphical user interface, on command line, or directly in R or Python. The involvement of different software in a complete DIA analysis and the different software versions often lead to an impaired reproducibility and robustness of the complete workflow, complicating the usability in general. Some analysis steps require programming skills to adapt and adjust as needed, limiting the accessibility to computational competent researchers. The complex data, in combination with a large dataset, require vast computational resources, limiting the accessibility and usability to advanced computer infrastructures. To overcome those limitations, we decided to integrate all crucial analysis steps into the Galaxy framework. Galaxy is a web-based workflow editor which offers a large number of tools, all with graphical user interface, facilitating the usability tremendously. Each tool is available as a Conda package and biocontainer and is open source. All analysis steps are started within the web browser and run on cloud and HPC infrastructures, circumventing the requirement of own computational resources. All analysis steps are stored within data histories with the respective parameters and tool versions. Individual tools can be connected and integrated into standardized workflows. Analysis histories and workflows can be shared and published via links and via mail. In total, Galaxy provides over 6,000 cores, 28 terabyte of RAM, and includes over 2,500 tools up until now. By implementing all the necessary analysis steps into Galaxy, we were able to generate an all-in-one Galaxy workflow for the DIA analysis. All crucial analysis steps for a complete DIA analysis are available in Galaxy. Furthermore, the modularity of the individual analysis steps allows to execute, skip, and redo parts of the analysis. Using pre-built and customized workflows allows for standardized and fast analysis of many experiments. To illustrate the functionality, I divided the complete DIA analysis into two workflows. First, the spectral library generation based on prior DDA measurement, which are first analyzed using MaxQuant and subsequently a spectral library is built using DIA-PySEF and open source tools. This example also includes a filtering step to only allow for unique peptides to be present in the resulting spectral library. In the next step, we can use the freshly built spectral library and retention time alignment file and the acquired DIA files in MZML format as an input for the open source workflow. In this step, peptides are identified and subsequently are scored using PyProfit. After applying the score on peptide and protein level, the results can be exported and further processed during statistical analysis. This workflow has been published under the shown link. And with this, I would like to thank you for your attention and special thanks to all collaborators for their effort in this project. Without them, this project would not have been possible. And now I'm looking forward to your questions.